Hello biologists, we are now going to start Mendelian genetics part 2. So we're going to jump all the way to slide number 48. Now we're going to begin crossing more than one trait in our plants. We did one trait at a time when we were doing things like earlobes and also when we were looking at pea plant colors and pea pod colors, but now we're going to cross two traits at one time. In this case, I'm showing you flower color and plant height. And these sort of crosses are known as dihybrid crosses. Di in science means two, hybrid meaning that we're actually mating two individuals. And so really what we're going to be tracking now is the outcome of two different crosses. We're going to use the same terminology we did in the last lecture, that is between being dominant and recessive and being homozygous and heterozygous. Here's our example here where we're going to be crossing a plant that is both purple, has purple flowers, and also tall. And we're going to assume when we first start doing this that we are beginning with true breeding plants again. So we have a true breeding purple tall plant. And we're going to be crossing that with a true breeding white short plant. The way that we distinguish between purple and white and tall and short is by using either capital letters for our alleles or small letters. And so we have big A, and big B over here to indicate that it is homozygous dominant for both purple and tall, and then little a, and little b for a plant that is both white and short. So you can kind of see how these are both true breeding. That is to say, even if we use the same rules we did in the last lecture, if you cross a plant that's purple with another plant that's purple and they're both true breeding, they will only ever produce purple babies. Likewise, if you have the plant be tall, mating it with another plant that's tall, and you do that thing a dozen or 50 or 100 times, they only ever spit out tall babies. So we know these are true breeding for both purple and tall. We therefore know their genotype, right? So we're going to go ahead and do that cross, and we're going to see what happens. Of course, remember, it's not the plant cells themselves, the ones that make up the flowers or the leaves or the height of the plant, that really is what we're talking about. We're talking about what happens in the gametes, in the sex cells. In the case of animals, that would be the sperm and the eggs, but in the case of plants, it would be the pollen and the ovaries. So let's go ahead and jump forward here. What are the possible combinations now if we were to mate an individual that is homozygous dominant for purple and tall with an individual that was homozygous recessive, that being white and short? Well, there's really only two possible gene combinations, okay? And I'm going to use my little symbol here for both sperm and eggs. And in this case, let's just say that the individual up here that's purple and tall is the male, while the individual that is white and short is the female. The only possible outcome that can be contributed to the gametes for this male is a big A and a big B. It's not even possible to contribute a small a and a small b because he's a true breeding homozygote. Over here, it's the exact same thing. All that this individual can contribute is either a little a or a little b. It's not possible to contribute a big A or a big B for this individual. That's what makes it a homozygous recessive true breeder. So if we were to take this sperm and this egg and put them together to return this organism, the offspring that emerges from this, to being 2N, that is to have two full complements of DNA, then what would the outcomes actually be? So let's go through, again, an F1 hybrid. Here we have an F1 hybrid where we're taking those two individuals, and all the plants are going to end up displaying the dominant form. Okay, and why is that? Well, we are going to have to do a full-blown cross here. And remember, in a true breeding individual, they can only contribute an A or a B. So all of the sperm are going to look like this, and all of the eggs are going to look like this for the female who is a true breeding short and white individual. See this? So that when these gametes get together, when they actually form a fertilized zygote, this is the outcome, right? So if you look at all of these outcomes, all 16 of these outcomes crossing in this cell here, we're going big A combining with that little a, that big B with that little b, just like that. And remember, A is for one trait and B is for another trait. And if you look at all of these, they're all identical, right? So that means that when you have a true breeding homozygous dominant individual mating with a true breeding recessive individual like this, all their offspring will exhibit the dominant trait. Remember, because as long as you have one capital there, that's the one that's going to be expressed. But all of the offspring are also carriers for the recessive trait. So it may not be expressing that recessive trait, but it's still carrying it. So this is kind of key here. This is just another way to look at that. Here we have our true breeders. They can only contribute this in their gametes, this one over here, this one over here. But when you put them back together so that they have the full complement of DNA, like their parents did, well, now you have your F1 hybrid offspring are all purple flowered and tall, even though they are carriers for the recessive trait. So what about the F2 generation? Let me back up one slide. What if we were to take all these individuals here in the F1 and figure out how many different varieties of sperm and eggs these guys would be able to produce, or in this case pollen and ova? It's going to look something like this. What is the possible combinations? So we're crossing that with that. Well, okay, what are the various combinations of things that you can get out of this one here? Well, you can take this A, you can combine it with that B, or you can combine the big A with that B. See that? You can also combine this little a with that big B, but you can also combine that little a with that little b. So, and then the same thing is with this one over here, the female. But take a look at this. It means that there are actually 4 different potential combinations of gametes that can come from this. Remember, what we've done is we've cut the amount of DNA in half, right? We had to cut the amount of DNA in half so that our gametes, when we put them back together, when we actually have these guys inseminate each other, that you will go back to having the same amount of DNA as their parents have. So when you do a dihybrid cross this way, you have to account for all 4 possible outcomes here. And it looks kind of like this. So, remember, these up here, this AB, 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 and AB, are all 4 different combinations that could exist in one individual's gametes, and these one over here in the other individual's gametes. So that when you cross them, they end up looking like this. Really complicated, right? Well, let's start with this first one over here. Look, big A combines with that big A, that big B combines with that big B, and this is what you get. So at least one individual here is going to be homozygous dominant for both traits. This big A combines with that big A to get these 2 right here, but this little b combines with that big B to give you this combination. So what about the genotype of this? Well, the genotype is that it's homozygous dominant for this, but it's heterozygous for trait B. However, the phenotype for this individual is that it will still appear the same as the one here to its left. That is, it will be both purple and tall. Okay? And you can do this little exercise here with each one, and then you need to be able to figure out what is the genotype of each one of these, what is the phenotype of each one of these, and then can you figure out the ratio of all of those individuals? Well, yes, you can. So what is the phenotype? This is how you figure it out. If you actually conduct this cross and you were to count all of these here on slide number 56, then this is what you'd get. You'd have 4 different phenotypes. You'd have 9 out of those 16 squares would actually turn out being tall. That is, at least one A would be there, if not both. And they'd also turn out to be purple. That is, at least one B, if not 2, would be present. So 9 out of those 16, tall and purple. 3 out of the 16 would be tall and white. 3 out of 16 would be short, or dwarf, and purple. And only 1 out of 16 would end up being short and white. okay, or dwarf and white. Now, if you want to check your math here, you can kind of add all four of these up. 9 plus 3 plus 3 plus 1, that equals 16. So we know that we've accounted for all of them. Now, if you want to pause the video here and actually back up to slide number 56 and make sure that these numbers look correct to you, now would be a good time to do it. But let me go ahead and jump forward and use a color-coded example of what that might look like. Remember here that these three here are dominant for at least something, but only one occurrence here, this one and this one down here, one for each individual, is going to potentially lead to 1/16 being homozygous recessive for both traits. So that 9:3 to 3:1 ratio always seems to hold for these kind of dihybrid crosses where the two traits are independent of each other. So in this case, we can count it up: 1, 2, 3, 4, 5, 6, 7, 8, 9. That's where we get our 9 that have the trait of being dominant for both, or exhibiting the dominant phenotype for both. Then we have 1, 2, 3, 3 being showing the dominant phenotype for A, both big A, little a, and big A, both display that. And then these three individuals here showing the dominant trait for B, but recessive for A. And then one individual being recessive for both. Now, this only works, and I had just mentioned this, and I don't know if many of you caught this, but this only works if A and B are independent of each other. Now, what do I mean by independent? I mean that if A and B are not located on the same chromosome. Now, we're not going to go into the details of why this is important, but we're really assuming that A and B are assorting independently on the various chromosomes within these organisms. If they were not sorting independently, for example, let's say that the gene for A and the gene for B were literally right next to each other on the same chromosome, the likelihood that crossing over is going to occur and separate those two is very low. Those two are probably going to be linked together for a very long time. We can't have that happen in order for our math here of 9:3 to 3:1 to work out properly. So, when Mendel was doing these crosses himself, he was lucky that those 7 traits that he was looking at tended to assort independent of each other. Let me give you an example of how this might work. And I've actually shown this to you before, the theory of independent assortment. Remember how you have your homologous chromosomes here? So here you have chromosome 1, chromosome 2, and chromosome 3. All right, so in these individuals, we're assuming they have 3 pairs of chromosomes. Of course, you have 23 pair, correct? So these are 4 different ways that you can independently assort or align these homologous chromosomes in meiosis. You can align them this way so that these light blue ones here, let's say they all came from your mother and the dark blue ones all came from your father. Or they could just randomly align this way, okay, on that cell equator. They can align this way or they can align this way. Now the thing that you need to remember here is that there's also the mirror image of all of these. It is possible to get all of these light blue ones to all be on the bottom and the dark blue ones to all be on the top. So even though we're only showing you 4 here, actually there's 8 different ones. There's these ones plus their mirror image. And so alleles end up assorting into gametes independently in these kinds of conditions. So for example, in our plants, when we're going with color and height, right, the color of the flower and the height of the plant, we were assuming that color is on one of these chromosomes, like let's say chromosome 1, but height, the information on how to make height, let's say it's on chromosome 3. So these chromosomes are independently assorting on there, and there is an equal chance that they're going to just get shuffled from one to the other and get pulled together. So this has to happen. This has to happen. And why? Well, again, if you had color and height both on chromosome 1, then they would never ever be assorted independently of each other with any other chromosomes, and the only thing that would ever separate them would be during crossing over. And if these two traits are basically neighbors next to each other on that chromosome, then the crossing over event would actually have to, the split would have to actually happen like perfectly right in between these two alleles. And that just is very uncommon. It's not going to happen. So again, our math for this only works if the two traits we're looking at reside on different chromosomes from each other. And just to kind of remind you guys of this, this is that slide that we initially brought in here from meiosis, but now I'm throwing in numbers at the bottom too. This is why it is there is a 1/4 chance of having these two alleles combine with each other. Remember, you got one from your mom, one set from your mom, and one set from your father here. And they have, in this case, let's assume there was only 2 chromosomes in this individual. It could align this way, or it could align this way. Okay? And each one of these outcomes, you could have a big A, big B here that are both like this from your mother. You could have a big A, a big B that align like this from your mother. Here's another 2 combinations from your father, and these are 2 combinations from your father. So it has to be this way. It's 1/4, 1/4, 1/4, and 1/4. Now, these are the 2 traits that we happen to be looking at, but realize there are a gazillion traits. There are tens of thousands, if not more, traits in your DNA that are all kind of assorting. And if you have a trait that's on chromosome 3 and you were actually crossing it and considering another trait on chromosome number 7, well, that's okay because they're assorting independently. But not all chromosomes are going to be independent. Obviously, there's multiple genes on the same chromosome. So our numbers are not always going to work out. And like I said, Mendel got lucky. I'm going to come back to that in just a second. All right, now let's back up and really think about what Mendel's contribution to all of this was. He publishes his work in 1865, and really very few people notice it. And the reason why is because, you know, he was way up in the highlands, separate from everybody, and they were essentially writing all of this stuff by hand. It's not like they were photocopying this stuff en masse. So if you wanted to have a copy of a textbook, you either had to have a printing press or you had to do it by hand. And distribution of information in the 1860s was not like it is today. So it really took until the 1900s that Mendel's work ended up getting rediscovered. And so his work ends up describing the behavior of chromosomes during meiosis. And this is really kind of critical because he figured this out mathematically, even though he had no clue what a chromosome was. He had never heard of the word meiosis. It had never been invented. And yet mathematically he figured out that you are getting something particulate from your mother and something particulate from your father. Right? We happen to know them to be genes on chromosomes. And that there's some assortment going on here that is allowing our math to work. And his math keeps telling him this, but he didn't actually know the mechanism of what was going on. That was not discovered until much, much later. Now, let's go ahead and do a little bit of a quiz. This might be something that would appear on your midterm coming up. An individual with genotype big D, little d, big E, little e, will have what combination of alleles in its sperm? Now, I'll let you pause and think about that for a second. But if you chose all of the above, then you're going to get that question right on the midterm exam. All right, now let's go ahead and move forward here so that we can understand why it is that the answer is all of the above. If you were to take this individual right here and figure out all of the different combinations that you could make into this individual's sperm, well, that big D could combine with that little e, or I'm sorry, with that big E, just like that. That big D could also combine with the little e, like that. This little d could combine with the big E, and that little d could also combine with that little e. Looks like that. So the answer is all of the above. Now, again, I want to reiterate to you, Mendel got lucky. And we often say that he got lucky because we look at the data that he presented, and they always seem to hit that 9 to 3 to 3 to 1 ratio. Or if you remember back to the first lecture, when he was only dealing with one outcome, when he was only dealing with one trait, it always seemed to be 3 to 1, right? It was always 3 to 1 in that F2 generation. And now, does that mean that he didn't have erroneous data sometimes? That he didn't, find some traits that didn't seem to behave correctly? No, of course he did. He just didn't write those data up. He only wrote up the stuff that ended up producing an exact 9 to 3 to 3 to 1 ratio. I guess the reality is we will just never know, because the man's gone and all we have is what he wrote. But because Mendel didn't really understand what was going on, he only cataloged those traits that he said, quote unquote, were behaving normally. Mathematically, they were behaving normally. And the reality is, though, that there was a whole lot of variation that did exist in his pea plants. But anytime he came up against a situation where there was a lot of variation, he simply didn't record that information. He only recorded the stuff that tended to work. Now, let's talk a little bit about the relationship of dominant alleles. If you have complete dominance, which is basically all of the cases we've talked about right now, then you only exhibit a single trait. For example, red versus white. Let's say the big letter, big Q, is red and little q is white. If you have one or the other, then your color is automatically determined. But this is not how all genes behave. Some genes are what are called incomplete dominant. And incomplete dominance means that in the heterozygote case, let's say where you have big A and little a, instead of only expressing the big A heterozygote actually exhibits a third phenotype. So for example, let me give you an example. If you have one gene is for height, where you're really tall, and in the case of the other one, you're very short, having a heterozygote case where you are have a big A and a little a, for example, you would have medium height. Okay, so that does occur, and when it does, we call that incomplete dominance. A third condition here is called codominance. In codominance, no one allele dominates the other. So, for example, both end up getting expressed equally. In fact, in some ways, they actually act independently. And we're going to talk about blood typing in a few slides, but that's the classic example of codominance. Now, let's go first to incomplete dominance, though. Incomplete dominance is where the intermediate or heterozygote condition produces some in-between trait. In this case, I've got red flowers as being homozygous dominant, white flowers being homozygous recessive, but when these two get together, the heterozygote condition produces pink flowers. And here's kind of how this works. So red flower plant times a white flower plant. In the F1 generation, you produce a pink plant. But there's also an interesting case. There's an interesting outcome when you talk about what happens in the F2 generation. So check this out. We have our red flowers there, 2 alleles, mark a red pigment. White flower means you have 2 mutant alleles or 2 recessive alleles. You can't make red. But in this case, a pink flower develops when you have 1 quote-unquote normal and 1 mutant allele. So essentially what you're doing is you're making less red pigment. If you were to look at this graphically, this is what it would look like. There's your parent generation, and you cross them together and you end up with your F1 generation being pink. However, what happens in F2? What if you took pink individuals and you crossed the pink individuals? Well, let's do that. If you cross the pink individuals, so here we have C sub R for red and C sub W for white, and the pink flower has this C sub R, C sub W. Well, now you're going to sort these into the gametes for this particular flower, and this is what you get. So here's C sub R, C sub W, C sub R, C sub W. And when you get the R's together, all of a sudden you get red flowers again. But these C sub W flowers and a C sub R means you get some pink also, but a W and a W means you get a white flower. So let's follow this thing through. In one case, I'm starting out with red and white flowers. They're making pink flowers in the F1 generation. And then in the F2 generation, my pink flowers lead to some red, some pink, and some white in a ratio of 1 to 2 to 1. Right? So that's a classic case of incomplete dominance. Now let's talk a little bit about codominance. And to introduce this topic, we need to go all the way back to one of my favorite movies, and yours, I'm sure. This is a total early 1980s movie, The Blue Lagoon, with Brooke Shields. Anyway, let's say that you're like this couple here, and you're on a desert island or a deserted island, and somebody gets hurt. Oh man, you need a blood transfusion. Maybe that's the guy, and he falls out of the tree, breaks his head, and he ends up having to need a blood transfusion. So here he is, but you know what? You're on a desert island. You don't have anything at your disposal, and you know, what do you need to know before you try transferring blood from one person to another? And so meanwhile, you're sitting out there going, oh man, I wish I'd listened in biology class. Here's what you need to know. You need to know your blood type. So before you go off to a desert island with somebody, maybe it's a good idea to know what kind of blood they've got. Because who knows, it might save your life. Now here's the genetics of blood typing. It turns out that you have 3 different alleles in your blood. In this case, you have type A, type B, and then type O. A, B, and O. Now, in this case, the A and the B indicate a particular protein that is found on the outside of your red blood cells. It's what your body uses to identify its own blood. So some of you have a gene here, or I'm sorry, you have a protein that is labeled A. Some of you have a protein that's labeled B. And some of you do not have either A or B. And so in the absence of A and B, we come up with O. Okay, and this I is simply indicating the gene. So here's the gene, and it puts out an A. Here's the gene, it puts out a B. And here you're lacking the gene. So instead, you are producing an O. And the gene that controls ABO blood typing, so A, B, or O blood typing, they end up coding for something called an antigen. And these things essentially identify you. If you were to take somebody else's blood and stick it in your body, it would be producing different antigens, and your body would totally freak out and essentially attack all those cells and try and kill them. Well, if you poured enough foreign blood into you with the wrong blood type, your body would essentially try and kill all those cells en masse, and it would be a big gooey mess in you, and it would probably cause you to die. So that's probably not a good idea. Knowing blood type is really, really important. Now, the reason why we're saying this is because type A and type B are both dominant. They're dominant, and so one does not dominate the other. And so when you have, if for example you have type A and B, then your body will be producing both antigens. And type O is actually recessive to either A or B. And what is the genotype for A, B, AB, and O? Well, in order to know that, let's go ahead and take a look at some of these crosses. Okay, let's say that this is you, where both copies of your gene are producing A. Some of you, and for those of you that are out there, if you know what your blood type is, that's fantastic. You can actually kind of keep track of this yourself. For those of you that don't know your blood type, it's probably a good idea to ask your doctor the next time you go and get a physical. But it is possible to be homozygous for type A blood. Homozygous meaning that on both copies of your DNA, you are producing A-type blood over here and A-type blood over here. It's also possible to be heterozygous for A. That is to say that you're producing A because of the genes of one set of genes that came from one of your parents, but the other gene that you got from your other parent is type O. Okay, let me skip over this middle condition and go all the way over to here to B. This is the exact same thing. You can either get 2 B's from both of your parents or just 1 B from one of your parents. In this case, you're getting neither, okay, that would produce type O. But over here is probably the rarest blood type of them all. In this case, you're receiving an A, and from the other parent, you're receiving a B. The most common blood type of all is actually O. The next most common would be A. And then B is less common than A, and then AB is the least common of all. And, this is really critical for understanding if you're ever going to undergo a transfusion. This is the first thing the doctor needs to know, because if they're going to have to be putting blood into you while you're being operated on, they need to make sure that they're putting the proper blood into you. And if your immune system ends up attacking the blood cells that you put into you, it could be very, very bad. So but then again, there's also something called a universal donor. Type O would be a universal donor. Somebody who has type O blood is not producing the A or B antigen. And so if they were to put their blood into you, regardless of what your blood type is, your blood would not reject it. It wouldn't reject it because it doesn't see either an A or a B on the surface of the red blood cells, and so it just accepts it. Now, the universal recipient, that is the person who can receive any blood type ever, would be somebody with type AB blood. So if you have type AB blood, then it means that you can receive blood from somebody with type A, you can receive blood from somebody with type B, and you can receive type O blood. All of it is going to be fine for you, and your body will see it all. And now the one other little factor, the one other complication to this is you need to know more than just A, B, or O. There's also a second factor involved here, and it's called the RH factor. I'm not going to get into too great a detail here, but it's just another protein, and instead of giving it a letter designation of A, B, or O, we say plus or minus. You either have it or you don't. If you have it, then you're RH plus, and if you don't, then you're RH minus. Now, we use these blood typing, so you might have actually wondered, why is it that I'm given a plus or a minus when my doctor told me my blood type? Well, they were giving you two things. They were telling you whether you were A, B, or O, and they were telling you whether you were positive or minus or negative for the RH factor here. So just so that you know, I know my blood type, I am A positive. That means that my cells are all producing the A antigen, and I also am positive for having this RH receptor on my red blood cells. So go and ask your doctor, find out what your blood type is. Alright, so let's move on to Mendelian genetics in animals, a little bit more of a practical application for this. In this case, let's say that pigment, the production of pigment, of color in these animals, is determined by a gene. This is more like a light switch. It's either on or it's off. And organisms that are capital C have their pigment gene turned on, whereas individuals with a little c, their genes are turned off. Looks kind of like this. So, for example, also genes for pigments produced, right? A or big A or little a. So we're talking about two different things here. We have a gene for pigment, and we have a gene for whether or not to turn that pigment on or off. And it looks kind of like this. Here's your A, that's your pigment for an allele. And if you're little a, then you're not producing any pigment. And you can be like this. Big A, or big A, little a, this is producing, this is a normal production of pigment. As far as your outward appearance would be, your phenotype, you would be producing pigment. But if you were in the situation where you were little a, well, you're not producing any pigment, and we would call that albinism, or an albino. Now, this means that other alleles that determine color, like for example, maybe there's a gazillion different alleles that produce color, but if you end up with a little a for this particular gene, it doesn't matter if you are if you have the ability to make those other pigments, this gene turns them all off, and so all other alleles for color don't matter if there is no pigment being produced. And this is a rare condition, but this is what it ends up looking like. Individuals that might otherwise look like this, when that gene is, when that individual has the albino gene, then you end up with individuals that look like this. Now let's go to another example here. Let's go ahead and do a dihybrid cross, and we'll consider two things. How about one's risk of developing albinoism or albinism, and one's risk of developing hereditary deafness? Let's assume that, and so we're going to ask ourselves, what is the probability of producing a child that is both an albino and is also hard of hearing? And what is okay, this is another way to state it. What is the probability of producing a deaf albino child? Well, let's go ahead and do the test cross, and we're going to assume that we've already started out with true breeding individuals, and for all traits, and then we're going to produce our F1 generation, and from that F1 generation, we're going to come up with all the various outcomes, the various possible outcomes that could occur in the sperm and in the eggs. And I've already put all of that stuff here for you. So here we have all four possible outcomes for these two different alleles. This one here, the A, is going to be our albinism one, and the D over here is going to be for deafness. So here we have an individual that is homozygous for the dominant trait of producing color and also having normal hearing. Here is an individual that is producing color but is deaf. Here's an individual that's not producing color and has normal hearing. And then here's an individual that is both not producing color and is also deaf. And this same combination up here, we are placing down here along this axis as well. And we can go ahead and do our cross. It looks like this. And so if we are asking ourselves, what is the probability of producing a child that is both an albino and also deaf? Well, it's only going to occur when you're crossing this gamete with that gamete, which is this cell right down here. And if you were to count these all up, there's 16 of them, so it would be 1 out of 16. You have a 1 out of 16 chance of producing an albino deaf child in this example. Now we're going to go ahead and complicate things just a little bit. Here we are making the assumption that a single gene controls for a single effect. But that's not always true. There are some genes in our bodies that actually elicit multiple effects. And so these are the different ways that I've listed here that genes can actually influence the phenotype. What is the phenotype? That's the outward appearance, right? So how does what is on your two sets of chromosomes affect what the outward appearance is? Well, this is the one that we've dealt with so far, is this top one, where a single gene ends up producing a single phenotype. That is, you have a big A and your flowers are purple. You have a little a and your flowers are white. Okay, that's straightforward. But there's also the case where a single gene can produce multiple effects. Okay, and we're going to talk about a couple of really good examples in the next few slides. Then there's also the case that if you have multiple genes interacting with each other, so we're not just talking one gene, but actually a couple of genes working in concert that produce a whole myriad of different phenotypes. And that certainly does happen as well. Now, the classic example of a single gene having multiple effects, we see that in sickle cell anemia. This case here is known as pleiotropy. And it's where an allele at a single locus, at a single location on your chromosomes, can have an effect on two potential different traits. And in the case of sickle cell, this is actually caused by a mutation where a single amino acid is out of place. And when that little amino acid is out of place, then the entire hemoglobin molecule, which is a major molecule part of your red blood cells, it totally misshapes the red blood cell. These little round ones that you're looking at, these 3 round ones, are normal red blood cells. But when you have a sickled cell like this one, it is because you have a small mutation in your protein sequence that makes your hemoglobin. And so you end up with an abnormal red blood cell. In this case, it is a single gene. It is a single allele that is ending up producing 2 potential effects, 2 potential traits. And so that's pleiotropy there. Now, I'm going to give you another really cool example of this. So these are not my puppies, but they sure are cute, right? Now, take a look at the colors here. We have a black lab here. We have a yellow lab, or also called a white lab. And then you have your brown or chocolate lab. So you've got these 3 different colors here in their coats. How do you get these 3 colors? Well, it turns out it's controlled by just 2 genes. And I should say that we're going to go into the details of how this happens, and you need to pay close attention to it the next, I think it's 4 slides or so, because this will be on the next midterm. I'm telling you now, this example will be on the next midterm. Now, when you have 2 genes interacting, that is to say, you have one gene that's controlling for one thing, then you have a second gene that is essentially turning off the function of the first gene. Well, this is called epistasis. That's epistasis, where you have genes interacting with each other. That's what epistasis means. So one gene can actually mask the expression of another gene. And again, it's like having a light switch, and where in one case you can turn a gene on or off, but in the other case you can actually turn off the first gene. And sometimes you will end up with a situation where the outcome that you thought was going to occur doesn't happen. And so epistasis really is all about the interaction among products of 2 gene pairs. And the classic example of this is hair color in labs. That's why I wanted to show you that picture first. Let's start with gene number 1. In gene number 1, you either produce 2 different colors. You produce black, you produce this black color here, if you are homozygous or heterozygous for the dominant trait, in this case black. If you are little b, or if you are homozygous recessive for this gene, little b, this is the only way that you will get a brown individual. Okay? So black individuals look like these 2, brown individuals look like this. All right, how do you get the white gene? Or I'm sorry, how do you get the white puppy here? Well, a second gene is responsible for the deposition of pigment, that is actually putting pigment in. If you are, these conditions right here, either homozygous or heterozygous for the dominant condition where you're actually laying down pigment, well, that's when it allows gene 1 to operate. However, when you have this condition down here, homozygous recessive, little e, then it doesn't matter what combination you have up here. It's like a light switch being turned off, and you're not producing any pigment at all. It doesn't matter what gene 1 is saying. Gene 2 can turn gene 1 off. In this condition, you get a white coat. Okay? This is how you get the 3. You want to get black and brown? You need to have this condition here where you have at least one big E present in order for these to be expressed. If you don't have a big E present, you have 2 little e's, well then it doesn't matter what you got up here, you're not going to get expressed. So what happens if we cross 2 heterozygote individuals for these 2 traits? That is to say, we took true breeders, we bred them together, we ended up with an F1 generation that was heterozygous for these 2 traits. Okay, big B, big E, little e, and you're crossing at times each other. Are they all going to be brown? Are they all going to be black? Are they all going to be yellow? What, or some combination? Well, the only way to do it is to figure out what your sperm and eggs are going to look like, in which case your sperm, let's say this is the male over here. In this male is going to be producing sperm that has big B, big E represented, big B, little e, little b, big E, and little b, little e represented in their sperm. And so you would put those along one axis, and then the same thing over here, you'd put that on the other axis to set up your Punnett square. So here we are again with our dihybrid Punnett square cross. And lo and behold, look at this. We end up with all kinds of different color patterns. So this is really interesting. We get ones that are homozygous dominant for both traits. We get heterozygotes throughout here, and we even get a few representing the recessive traits as well. If you were to actually look at which ones were black, in order to be black, you have to have at least one big B present, right? This individual is going to be black. This individual is going to be black because it has at least one. But in order to express this black, these individuals that have black also have to have at least one big E present. That is turning it on. This individual does have the black gene. However, it also does not have a big E gene, and therefore it's not going to turn on the black gene to begin with. See that? So this individual here ends up being white. Same thing here, same thing here, and same thing here. Any place where you do not have a big E, you're going to be white. And any place that you are, and if you're going to be brown, it can only occur when there is no big B present, but there is at least one big E present. So it ends up looking something like this. Unfortunately, I had to invert the colors here. These white ones here mean black. It's just that it didn't turn out that way. My apologies. So 1, 2, 3, 4, 5, 6, 7, 8, 9. We end up with 9 black individuals. 1, 2, 3, 4 white or yellow individuals, yellow labs. And then 1, 2, 3 brown individuals. Now, can you reproduce this on an exam? If you can, then you're going to get this question right on the exam. Now, I'm going to back up to slide number 87 here. I just want you to let me go back one more, or even one more from here. I just want you to remember what the outcomes are, what gene 1 controls, what gene 2 controls, and how you arrive at each of these 3 different color codes. Then you need to remember how to develop these individuals here, okay, so these 2 individuals, how you're going to develop the sperm and the eggs from these individuals to make your haploid cells, and then put them back together in this example here to produce all of your outcomes, and then what the ratios are. Out of 16, you end up with 9 blacks, 3 browns, and 4 yellows or white labs. Okay, and you can add them all up, and you should end up getting 16. Okay, let's go ahead and add one little complication to all of this. So far, all of the genes that we've been talking about, the phenotypes, the phenotypic outcomes, the outward appearance of these single genes has always been one phenotype. One gene leads to one phenotype. That was true whether we were talking about flower color, the height of a plant, the shape of a red blood cell, or your earlobes, or whatever. These are all single genes where it's a one or the other. It's an either-or kind of situation. These are known as discontinuous variation. That's everything up until this point has all been discontinuous variation, where a trait ends up exhibiting only one phenotype or another. For example, pink or white, tall or short. In fact, I have a bunch of good examples here. Where you do a test cross, and what you get in the babies and the outcome is they're all going to be either tall or they're all going to be short, or some are going to be tall, some are going to be short. You don't get any intermediates. You're either all of your pea plants are going to be either wrinkled or smooth, some combination in there, but you're not going to have any individuals in the offspring that are something in between wrinkled and smooth. You know, you count up all of the babies in your pea plants and you crack open their pea pods, and they're either going to be green or yellow. They're not going to be something in between. And so, but this is unrealistic, right, for all traits. Only some do this. And again, this is another reason why Mendel got lucky. He ended up choosing traits that not only disassociated on different genes, but they were also discontinuous in nature. They were either one thing or another. But again, not everything follows this. I mean, just imagine this example right here. What if we measured the height of all 14-year-old individuals out there? Would we end up with two groups like this? Let's say that we went to whatever the local school is next to where you live and measured all the 14-year-olds. Are they all going to be 6 feet tall or 5 feet tall? No, of course not. They're going to look kind of something like this, right? They're going to have a range in variation. Most of them are going to be kind of in the middle, but you're going to have some off to the extremes of slightly short or slightly taller, right? So that goes to show that whatever controls for height, it's not just a discontinuous gene thing where your babies come out and they're either 5 feet tall or they're 6 feet tall. That would be an example of it being discontinuous. Or in the pea plants, the outcome of the pea plant crosses is you either got a tall plant or a short plant. That's discontinuous. So now we're going to go ahead and add a little complication to this in that we're not going to be using discontinuous genes anymore. We're going to use something called continuous variation. Continuous variation. And this is a more realistic case for some of the traits that you're familiar with. For example, hair color or skin color, these sorts of things. Where skin color, for example, ranges. There's a whole variety of ranges and phenotypes that you can have. And the height, great example. You take all the individuals in our state, and they're all going to range from tall to short. They're all going to range from dark to light, or heavy to not heavy, or light. And that's because these sort of traits, okay, we're not talking about pea plants here anymore, or flower color. We're talking about things where there's a lot of variation. And they all tend to fall into what is known as a normal distribution, with the majority of individuals falling in the middle, and the frequency of the extremes kind of tapers off on either side. These are extreme phenotypes here on the ends, like people that are extremely tall, over 7 feet tall, versus people that are extremely short. That is, they're less than 4.5 feet tall, let's say. And so this normal distribution is pretty much what we're talking about here. That's the continuous variation part of this. IQ is another good example. The vast majority of people fall right in the middle, but what controls IQ is actually variable. There's a whole bunch of different variables. On one end, you got this guy from one of my favorite movies, Revenge of the Nerds, and on the other extreme, you got these guys. Yeah, how about that? Here's another important one. Average newborn weight. For those of you that actually have children, you'll know what I'm talking about. The average weight of a child when a child is born normally. On the low side, probably somewhere around 6.5. On the high side, somewhere around 8.5, right? That's kind of a normal range. And so the vast majority of newborn babies that are born normally are going to fall into that range. But on occasion, you will get some individuals that are on the smaller side and some individuals that are on the taller side. But, those are the extreme rare cases that you get to hear about sometimes on the news. But for the most part, the distribution is like this for the weight of babies as they're born. On one side, you got little babies, and on the other side, you got big babies. And, all right, so this is an example of continuous variation. From small to large, or from dark to light, or that kind of thing, or IQ from not so smart all the way up to pretty smart. So how do you explain continuous variation, though? I mean, up until now, we only have familiarity with a single gene kind of controlling things, or at least two genes kind of interacting. But, if we have a single gene and that doesn't seem to explain it, then how can we explain the range of phenotypes? How do you end up producing it? Well, this is a really good classic case. It's called polygenic traits. Poly means many. Genic refers to your genes, right? So you have polygenic traits are ones that are the result of multiple genes. Traits caused by two or more genes. And each gene ends up contributing to the phenotype, and it compounds itself. So in this case, let's say that we have 3 genes here. Okay, see these 3 here? And that it's not so much dominance or recessiveness, it's how many of them are actually turned on in this case. So here you have an individual that we're saying has a characteristic of being completely white, and all 6 of these chromosomes that they have in this individual here are turned off, whereas in this individual they're all turned on. See that? And we're going to cross these individuals, and in the F1 generation, that's where you get your heterozygote. This is somebody that's heterozygous for all 3 traits. And you'll notice that the phenotype, the color outcome of these individuals is somewhere in between these. But when you take these individuals and then you cross them, you end up getting all of these different variations in here. Everything from completely white to this individual right next to it with only 1 gene turned on. This one's got 2, this one's got 3, 4, 5, and 6. This is the darkest case over here. And so you end up with individuals that fall into this range where the vast majority of them are this intermediate phenotype. But you still end up expressing the extremes as well. And what's really weird is that the environment can also affect this. This isn't just your genes. I mean, you can think of a time when, in the year, when your skin tends to be darker than at other times of the year, right? Obviously, if you're out in the sun in the summer, hopefully you're wearing sunscreen, but even if you are wearing sunscreen, you're going to get a little bit of sun and your skin's going to darken a little bit. So it's interesting that your genes kind of determine your base coat, but that how much sun exposure you have ends up kind of varying that. But this is just another way to look at it. This is identical to the image that I showed you before, with the exception that we actually are looking at the physical crosses here between sperm and eggs. Okay, you will never have to reproduce this on an exam, so don't worry. Slide number 97. Just know it conceptually. In fact, just knowing slide number 96 is probably fine, not having to do this entire cross here. So what is the results? Well, you can end up getting a large number of genetic and phenotypic combinations when you have multiple genes interacting. And so each gene ends up compounding, it ends up adding in a compound fashion to the phenotype. And the effect of individual alleles can actually be kind of small, but when you consider them all together, they end up affecting things greatly. Let's go ahead and use height as an example. In this case, let's just assume that we've got our 4-gene condition here, right? If you have all 4 of them turned on, then you're really tall. You're 6 feet tall. If you have only 3 of the 4 turned on, well, then you end up being 5'9". Here we have 3 turned on. Here we have 2. Here we have 2, 2, 1, 1, and none. And considering this case here, here are all the heights. And if you were to actually plot these individuals, you still end up with what's called a normal distribution, just like we spoke about a few slides ago. So that's kind of interesting. And what this means is that 2-gene trait, even if you only have a 2-gene trait, you end up with 5 different phenotypic outcomes. And you arrive at those 5 phenotypes from 9 genotypes. And if you need to back up to slide number 99 to double check yourself here, we have a 2-gene situation here, okay, A's and B's, right? And they are producing 9 total outcomes. And these 9 total outcomes can be broken down into 5 different phenotypes. These right here are the genotypes, and the phenotype is what you would actually see in these individuals. Obviously, this 5'6" individual and that 5'6" individual, they have a slightly different genetic combination, but you wouldn't be able to tell that just by looking at them, from the outside, because they'd just be 5'6", regardless of what these actually were. So the phenotype exhibits a nice bell curve in 5 different traits, 5 different phenotypes that arrived from 9 different genotypes. Now, the most common phenotype ends up being midway between the extremes, right? Now, if you were to continue this and say, well, that's with a 2-gene trait, what about for a 3-gene trait? You'd end up with 7 phenotypes. What about a 4-gene trait? Well, 9 phenotypes, etc. It's interesting that actually some traits, like eye color, are controlled by a lot more than 4 genes interacting. In fact, it's controlled by dozens of genes, which is why there's more than 9 different eye colors out there. You have whole shades and variations of brown eye color and greens and different types of blues and all of that. And the reason why is because it's more complicated than that. You have multiple genes that are all interacting and influencing each other. And, well, what about skin color? Is that polygenic? Well, of course it is, right? However, as I indicated before, it's not entirely genetic, though, right? What else could be influencing skin color? Well, hopefully you remember that the environment can influence it, right? So let's just say that you're this individual here, and but you're in the sunlight, and so yeah, that's what we're talking about, right? Tan lines, right? Tan lines. So these are individuals here whose feet are expressing in one case what their genes are telling them, and in another part of this of the foot where the skin is exposed to the sun, that is being influenced. The amount of pigment that is being produced is being influenced by the sun. So these are basically multifactorial traits. This is the same plot that you saw before. I'm just plotting it with percentages over here. But don't worry about any of these numbers. Just remember that it's a normal distribution when we're talking about these multi-factor kind of traits. So multifactorial traits are really just traits from 2 individual or 2+ alleles that that are being influenced not just by the influencing each other, but also are being influenced by the environment. It's really, really difficult to predict the phenotypes in these cases because you could potentially have a genetic combination that produces a slightly lighter shade of skin, and yet you're the kind of person that hangs out in the sun a lot, so your physical skin color ends up being darker than your genetics would predict, right? Now, there are some instances where the environment really determines what the outcome is. Hydrangea flowers are a really good example of how the environment can truly affect phenotype. In this case, hydrangea flowers can range anywhere from like a bluish violet color all the way to pink and every shade in between. And what color the flowers actually exhibit is determined by the acidity of the soil. So you have very acidic soil, and the flowers end up taking on one color, and if the soil is less acidic, then it takes on the other. And if you change the acidity to something in between, well, then you get an intermediate shade of color. And I can't remember exactly which flower is which, but if you want to grow hydrangea flowers, I recommend looking it up on the internet and looking for how to do this. So what does it say? Well, it says that your phenotype is determined by your genotype, but also by environmental factors. You know, we managed to pick a few traits that are discrete, and that made it really easy to understand the genetics initially. Like, for example, our discrete traits included things like your earlobes, whether they were attached or detached. But, you know, there are a lot of genetic disorders that are also discrete. You have one gene, and it can produce a genetic disorder. The pea plant colors of the flowers or of the pods or of the actual peas themselves, discrete traits, right? But the reality is we also have continuous traits too. Our height, our weight, our eye color, our skin color. These are all things that are continuous traits. So we want to kind of make sure that we understand the difference between those. Now, the last subject that we're going to talk about, I'm actually going to leave up to you guys to learn more about this in your textbook, but it has to do with sex-linked inheritance. And this has to do with the fact that males and females, while we might be the same in 22 of our 23 pairs of chromosomes, that one chromosome that we have that makes us different also changes how we inherit things. Females have 2 X chromosomes, and males don't. We only have one X chromosome. Now the other little tiny fragment of chromosome that we have that takes the place of the other X, we call it a Y, but it's really just this really small fragment of chromosomal DNA. And so this means that if you have a genetic trait that gets crossed on the X chromosome, well then in order for a female to express that trait, sometimes it has to be present on both. On males, since we only have one X chromosome, it makes it much easier for us males to exhibit an X-linked trait. Really good example of this would be male pattern baldness. The gene that controls whether you go bald, it's on your X chromosome. And so that means that if a male gets an X chromosome with the baldness gene on it, then they're going to be bald. If a female gets an X chromosome with the baldness gene on it, it doesn't mean they're going to go bald because they might not have it on the other X. If they don't have it over here, then they'll just be a carrier, but they won't be expressing the baldness. That's why it's far more likely for males to be bald than for females to be bald. There are bald females out there, but you end up having to have that trait present on both X chromosomes in that case in order for it to be expressed, and that's rarer than to get it on only one X chromosome on a male. Okay. Oh, and by the way, just for those of you that were interested, there are a couple of chromosomes, a couple of genes that are on the Y chromosome and hence are therefore only present in males. One great example, ear hair. So for those of you that want to know why it is that females don't tend to get hair on their ears as they get older, well, it's because they don't have Y chromosome, and that gene is only on the Y. Alright guys, so read up on the sex-linked inheritance thing, and I will see you all next week.